Hey, my name is Phoenix Locklear, and today we're going to be discussing a podcast that is put out by bacteriophiles, and the title of this podcast is Globules Get Garbage Gone. This podcast is available for anyone on the American Society for Microbiology website, and I'll also include the link down below in my description. To begin this discussion, it is very important that there is a very good understanding of the two different types of cells. These two different types of cells include the prokaryotes and the eukaryotes. This presentation focuses primarily on the prokaryotes. Eukaryotes are more complex cells who contain various organelles who each have a specific role. Each organelle works together to keep the cell healthy and productive. The human body is composed of eukaryotes, and they are also found in plants, animals, fungi, and insects. I once had a professor explain to me that a eukaryote, or a working cell, is almost like stepping foot into a factory, and once you do, each organelle is doing its own specific job, and the ultimate job is to get the job done. On the other hand, prokaryotes are what makes up bacteria. They are very simple, single, or unicellular organisms. However, prokaryote cells include smaller, circular DNA that is held within its nucleoid, because if you recall that I mentioned earlier, only eukaryotic cells include a nucleus. As we continue talking about the various types of cells, this slide will provide you with an illustration of the different types of organelles that are present in both eukaryotic and prokaryotic cells. If you will notice, the prokaryotic or simple cell does not contain a lysosome. This is very important because on the following slides we will discuss how the prokaryotic cells pretty much develops its own lysosome. Lysosomes are responsible for removing waste from the cell and is often referred to as the garbage disposal and recycling center of the cell. As we continue this presentation, we'll begin talking about a specific type of cell, which is the prokaryotic cell. As you will notice, this slide will show you the morphology of a bacteria cell. Bacteria cells lack digestive and circulatory systems, so they do not have a proper way of disposing waste, such as unwanted metabolic products. Scientists have known for a very long time that certain bacteria are capable of producing smaller versions of themselves by literally breaking smaller parts of themselves off. Although the smaller versions of themselves do not contain certain materials such as DNA to reproduce, a recent study by the University of California, San Diego has shown that these mini cells are the bacteria's way of cleansing themselves of waste that is accumulated within the cell. The mini cells discharge damaged proteins bundled inside of it. The cell capability of developing mini cells is thought to be a survival mechanism. To develop the conclusion that bacteria cells were capable of creating their own waste disposing system, researchers used two different strains of the bacteria E. coli. The reason why the researchers use this specific bacteria is because it is known for these cells to create mini cells of themselves. So the researchers took a normal strain of E. coli along with a mutant strain of E. coli, and they did this to examine the various effects of mini cell production on the two bacteria cells. Both of the cells were introduced to a non-lethal dose of streptomycin, which is an antibiotic. However, the dose was strong enough to cause stress on the cells, which is all that's needed to create the initiation of mini cells. As the antibiotic damages the proteins of the bacteria, it causes the proteins to clump together. These strains were allowed to grow in culture, and then they were visualized up under the microscope. Once visualized, it was determined that the antibiotic exposure increase the mini cell production of the mutant strain of the E. coli more so than the normal strain of the E. coli. To determine if mini cell production and damage could be associated, the researchers studied the production of mini cells by old poles and old daughters of the bacteria cells. To give you a better understanding of what the researchers mean by old poles and old daughters, I have included the photo to my left. A rod-shaped cell has two ends, and when it divides in half, each new cell receives an old end from the original cell since they divide from the middle of the original cell and a new end. The old daughter is when a cell divides into two, and one receives the old daughter pole from the parent cell, and the other receives a new pole. So both daughters are the same age, however, one has a new pole and an old pole, and the other has the same, but the older pole is a little bit older. The researchers were able to determine that the old poles accumulated more damage and waste materials than new poles, and old daughter cells made more mini cells than the new daughter cells. Thus, the origination of mini cells appears to be linked with old poles and old daughters, both of which have been shown to accumulate damage at a higher rate and create more mini cells whether exposed to antibiotics or not. Following the determination of mini cell production and if the damages within the cells was associated, the scientists had a few more questions in regard to the mini cell production and how it actually took place. To determine this, the scientists wanted to determine if there was a link between the mini cells and the health of the bacteria cells. The researchers tagged and tracked the protein with fluorescent markers within the bacteria. While under attack from antibiotics such as streptomycin, the researchers used microscopy techniques to follow damaged protein within the E. coli bacteria to the pole or the end areas of the bacteria cell, and then they watched as it was packaged inside the mini cell and finally dispatched out by the bacteria. These new results described how natural the route was of the bacterium exiting the unwanted waste, and it also provides many leads with how to develop many different pharmaceutical techniques that can aid in the assistance of antibiotic-resistant bacteria. The elimination of waste through mini cells is such a big deal because it allows bacteria to grow a higher tolerance to certain antibiotics, and in this case it was streptomycin. And it also allows the bacteria to have higher levels of stress, which can cause more antibiotic resistance in the future. If this mechanism extends from streptomycin to other antibiotics, mini cell production could be an overlooked pathway that bacteria are using to resist antimicrobials. It allows bacteria to be healthier in the long run by getting rid of burdensome waste. It was also concluded that the cells who had to produce mini cells took longer to grow because they have to take account of the extra volume that they have. However, the offspring of the cells that made mini cells had a faster growth rate, especially in the presence of antibiotics. And this will conclude my assignment of the synopsis of the podcast Globules Get Garbage Done. This current slide will end my presentation. It will also include all the references and works cited in relation to the information I discussed. The first link will be the link to the podcast itself on the American Society for Microbiology website. It also will include the works cited reference for the article that goes along with this research. Thanks.